Survey says that we should go ahead from the point we left yesterday. So the question that was addressed to you yesterday, and a complete almost 23 hours time was given to you to ponder over the idea and to understand or to think about that what exactly went wrong in the experimental design that was used by Avery, MacLeod, and McCarthy, which made Hershey and Chase plan another set of experiments so that they can conclusively prove that it is the DNA which is the information bearer. So, can anybody elaborate upon or what kind of flaws that you can point out in the experimental design that was used by Avery, MacLeod, and McCarthy? Yes. Anyone? Should I give you the background of the experiment again? Okay, from the Griffiths experiment, it was. Who is that? So, anyone wants to give me some background related to Avery's experiment, and then we can move ahead from that point. Yesterday, we saw that from Griffith's experiment, it was understood that there is some kind of a transforming principle or a molecular factor that is involved in changing the characteristics which are associated with a non-virulent form of the bacterium to a virulent form. So if that can happen, even when you are using the heat-killed concoction of the virulent form of the bacteria, it was found that it was capable of transforming the avirulent form into the virulent form. So it was supposed that perhaps there is something which is responsible for bringing about this transformation. And the molecules or the molecular entities that perhaps are involved in bringing about this transformation can be of four different types. The nucleic acids, first are the nucleic acids, next comes proteins, then carbohydrates, then lipids. So yesterday's discussion, we somehow ruled out carbohydrates and lipids. And Avery's experiment actually focuses more upon proteins as well as nucleic acids. So what they did was that they used what you can say a homogenate or the concoction that was obtained from the S strain or the virulent form, and they tried to remove one of the two. That means either they removed the proteins by using the organic solvent extraction method. So they actually extracted out protein from this entire concoction. And they found out that nucleic acids perhaps are responsible. Then within nucleic acids, they use ribonuclease in order to digest RNA. And they found out that there is still the transforming activity retained with the concoction. So perhaps they found out that it is the DNA which is the information bearer, which is capable of transforming the avirulent form into the virulent form. That's what was the conclusion. So do you feel now that there is actually a need to go for another experimental design that can prove that DNA is the information bearer? So that was the question where we left our discussion yesterday. So what's your take on it? Did you try to think over the concept? If you look at it, that how they arrived at DNA being the genetic information bearer, then perhaps you would be able to make out what was the problem that actually made others to plan another set of experiments. Yes? Anybody? At least, can you see any one problem with the setup? Yes? How many of you actually tried to think over it, first of all? None of you? Okay, Sarvesh tried to think over it, but perhaps he hasn't got to any answer. Okay, one thing is during the experimental setup itself, there is a problem in the sense, first of all, either you are removing proteins by means of an organic extraction method, organic solvent extraction method, and when you do that, then when you remove proteins, you actually don't take out carbohydrates, right? In organic extraction method, proteins get precipitated out of the homogenate, but you are not sure whether it is getting out carbohydrates or not. It may also get out lipids when you are going for organic extraction, but carbohydrates still remain. So it could be between nucleic acids and carbohydrates. One point. Okay. You're not very certain that whether carbohydrate, that means whether carbohydrate could be the transforming principle, because although they are not as stable as nucleic acids or proteins, or they might be used as an energy source by other organisms, there is a possibility that perhaps these molecules still retain the capacity of bringing up transformation. So you haven't actually paid any attention towards the residual amount of carbohydrates which might remain in your concoction. That is one point that is to be addressed. Two, when you are using any of these methods, see, either organic extraction method, or that means organic solvent method, or you are using any of the proteolytic enzymes or proteases, you are not very certain that there will be a complete digestion of proteins. So there could be some residual proteins remaining, which are bringing about the transformation. So when you are using nucleic acids as perhaps the transforming principal source, you are not very certain that it is only nucleic acid. Okay. So when you use RNAs as well, then also you are not very certain that it is completely removing the RNA and only DNA is remaining. Okay. But one last thing is something which is astonishing of all these, and that is when you use DNAs, then there is a problem that you see. And that is that perhaps because DNA is getting digested, there is no transformation happening. But because of these flaws that the experimental design had, there was something that is like, see, you might have done this in your chemistry practicals, that when you are looking for some kind of an entity in your sample, then the normal process is that you first use what is called as a preliminary test, and then if you find out, okay, that there is some evidence that there is presence of this particular entity in my sample, then you go for the second test, which is called as the confirmatory test, right? Yes or no?
Yes, sir.
Okay, so likewise in Avery's experiment also, it was just indicated that there is DNA which is working as the genetic information bearer. There is just an indication. They haven't conclusively established the fact that, okay, when I use DNA, then whether I can bring about transformation or not. That was exactly what was done by Hershey and Chase in order to make it completely, what you can say, prudent for everyone to understand that, okay, if the DNA gets transferred from one organism to the other, then there is actually transformation happening within the organism. And what was Hershey and Chase experiment? If anybody can volunteer to explain, it's actually in front of you, but still, how was it planned? It's a classic example of how you should plan the experiment depending upon what is your aim, what is your hypothesis. On the basis of that, how should you plan an experiment? This is a classic example of it. So, so can anybody let others understand that what was exactly there planned in the Hershey and Chase experiment? Yes, anyone? Yes, anyone? Okay. No one remembers the Hershey and Chase experiment. Have you actually studied this experiment previously? I believe that you have. Yes. Yes. Does anybody remember Hershey and Chase experiment, or should I do the job of explaining? Okay, because nobody is coming up with experimental design. Okay, it was related to. It was known well. It was well known that when a virus affects a bacterium or infects a bacterium rather, then there is a chance that the viral genetic material is getting transferred because the virus uses the host as a, a factory for its production, right? So if it's doing so, then perhaps there is somehow it is transferring its genetic information to the host, and then that information is utilizing the host's machinery is used for production of new viral particles inside the host. So, keeping this in mind, Hershey and Chase planned another set of experiments wherein they used up the T2 phage, which is a lytic phage and infects E. coli. So the host was E. coli, and the phage that they used was the T2 phage. Now they did something which was quite a remarkable stuff. Now you have learned something about this in your isotope biology section, and that is what is exactly done here. In order to label proteins, what they did was that the entire viral particle proteins were labeled with S35. S35, that is sulfur. Why they must have used sulfur as the label? Can anybody give a view about this? So that why sulfur must be chosen as the uh, atom with the label for labeling proteins? Yes, any idea? Why only sulfur? Okay, there is something that is coming. Sarvesh, yes, you are using radioactive isotopes for labeling because that's the purpose behind the study, in order to track the molecule, whichever molecule you are trying to see. But why only sulfur? Why can't I use any other molecule within the protein, or other atom within the protein rather, not the molecule? Why should I use sulfur to label proteins? Any idea? Apply your principles of radiobiology or isotope tracer techniques that you might have developed during your studies of isotope biology. There's a basic principle of isotope labeling, or uh, the tracer technique, and that is that you should choose an atom within your molecule in such a way that it remains unharmed all throughout the process. Okay, so when you use sulfur, it will remain unharmed within the proteins only. Now, so there is an important aspect here. Now, sulfur, do you find sulfur in DNA? Yes or no? Is there any trace of sulfur in DNA? Yes, exactly. There is no trace of sulfur in the DNA, right? You never get sulfur in the DNA. So automatically, if I choose S35 as my label to label the proteins, then I am quite certain that the label will be essentially only remain with proteins. It will never go to DNA. So now the interesting facet here is that you just need to make out that which molecule is actually responsible for doing the transformation. Whether it is protein, you have only two choices now. So you have zeroed down on the basis of the experiments that were done by Avery, MacLeod, and McCarty. You have actually zeroed down from the 4 biomolecules to only 2. That means you are referring only to proteins and nucleic acids here. So you need to label only 2, correct? You just need to label nucleic acid and you need to label proteins. So proteins can be labeled by using S35. So S35 label goes along with proteins because there is no trace of sulfur in the DNA. And P32 label will go along with DNA because this is a T2 phage and it contains only DNA and proteins essentially, right? There is no RNA here. So now the choice was very simple. It was only proteins and DNA. So they labeled proteins with S35, DNA with P32. Now you may get some traces of phosphorus in proteins, but you will never get the trace of S35 in DNA. So in case there are some overlapping proteins which are responsible for bringing about transformation, perhaps they might get labeled with both P32 as well as S35. But if there is only proteins, only DNA, then you will get only P32 because you will never get both the labels in the DNA. Is that point clear? Should we go ahead from this point onwards? So is it now clear in everyone's mind that why they have chosen only sulfur for proteins and phosphorus for DNA? Now it was simple. Now they prepared 2 viral particles, okay, and one viral particle was having only its protein labeled with S35, and the other viral particle was having only its DNA labeled with P32. Okay, so now they carried out the infection process. Now, when it was allowed to infect the E. coli, then it was found out that now there is very what you can say well planned schematic here, that you can very easily do this. what you do is that you allow the viral particle to infect E. coli. That takes somewhere around say 2-3 minutes. And once those 2-3 minutes are done, then you can very easily remove the viral particles which are on the surface of the organism, that is the host organism, by using just a blender. Okay. So, and then you can check that what is there, which kind of label is there inside the E. coli. Okay. So infected E. coli cells were then traced for the label. So what do you think, what must be the label that must be retained by E. coli after infection is done? So you allow viral particles to go along with your host organism, that is E. coli, for 2 minutes, and then you put it inside a blender and you just remove, that means the leftover viral particles. And what you do is that you then check what is the extent of label inside E. coli, inside host. Okay. So what do you think, what must be there inside the host now? Yes? Anybody? It's there in front of you. Every process is given in front of you. There is a viral particle that infects the host. Then you put it inside a blender so that the viral particles are removed. You have only the host with the label. Which label do you suspect to be present inside the viral particle? Yes. So will you get S32, S35 label inside the viral particle, or P32 label inside the viral particle? Is the experimental setup understood first of all? Okay, let me find this out. Are you okay with the experimental design?
Yes, sir.
Okay, so what do you think? What must be the label inside the host?
Inside the host, P32,
Yes. So what does that indicate?
that DNA is being transformed.
Not transformed. DNA is being transferred from the viral particle inside the host. What it actually indicates is that it is the molecule which is responsible for transforming the organism into a viral factory, right? That will be only possible if you are transferring the DNA inside the host, right? And that is proven here completely. So do you find any kind of a flaw in this experimental setup now? What's your aim? Your aim is to find out whether the transforming material is DNA or protein. So you have very skillfully labeled the virus particle, all proteins labeled with P35, and all DNA labeled with S35, and all DNA labeled with P32, right? So is there any problem in this setup, and do you think that there is any other need in order to establish this fact that DNA is the transforming material?
No, sir.
Right. So this conclusively proves, this experiment, that DNA is the transforming material. Now, once you know that DNA is the transforming material, then there must be a process wherein this transforming material is actually relaying the information, right? And then came the establishment of the central dogma of molecular biology, which talks about that there is a process in which the gene is converted into mRNA molecule, and that mRNA molecule is further converted into protein or translated into protein. So this outline of gene expression that we already know about came up from these studies which were conducted much before. the central dogma of molecular biology came into existence. Okay, so there are 2-3 things here that I need to highlight, that before you predict anything like this, that DNA, okay, we have understood that DNA is the transforming material, and it is the information bearer that can carry the information from one generation to other or one organism to other. Now, if that is true, then how does the information gets executed? That's what is the essence of understanding gene expression. Okay, so, and why do you find that there is a need for putting up this idea that there has to be a relation between the gene and the protein? Why there was a need is because of the establishment that was done much before understanding that DNA is the transferring material. It was in 1902 when a physician called as Archibald Garrett actually gave up the foundation that there must be some link between the transforming principle, which was already previously, there were studies that were done by, if you remember, there were some genetic studies that were done by Mengele. And on the basis of that, there was something that was established, that there is a hereditary unit which is responsible for carrying the information from one generation to other. And that hereditary unit has some relationship with the phenotypic expression. So on the similar grounds, Archibald Garrett established what you can say, you can consider this as a hypothesis only at the time. But he used up a very interesting, what you can say, uh, interesting analogy here. And that is, he was studying a human disease which is called as alkaptonuria, in which you actually excrete a compound which is called as homogentisic acid in urine. And when you are excreting homogentisic acid in the urine actually turns black. So there is this peculiar character that you can see in those humans which are affected by alkaptonuria. This is a genetic disorder, and the interesting thing that was proposed by Archibald Garrett was that is some kind of a metabolic, what you can say, disturbance that is created in the metabolic pathway. And because of that, what happens is that the person who is affected with alkaptonuria is unable to degrade an amino acid which is called as phenylalanine. And because it is unable to degrade phenylalanine completely, a precursor remains accumulated in the system, and that is homogentisic acid, which is responsible for black color of urine. Now, because this character or this disease condition is transferred from one generation to other, Garrett reasoned it, that there must be some involvement of a gene which is getting transferred, a hereditary unit which is transferring this information from the parent to offspring. And because now the gene is transferring the information, the gene must be responsible for production of the enzyme, which is one of the factors involved in phenylalanine degradation. See, it is being predicted in 1902, when there was no background of this gene expression overview that I'm giving you, right? So you can understand that how much foresighted Archibald Garrett must be, right? So on the basis of this background that was given by a study conducted by Archibald Garrett in 1902, and on the basis of all these establishments that we have done now, where we prove that DNA is the information bearer, later on there was an establishment of a relationship between genes and proteins. So before we go ahead into understanding that how this relationship between genes and proteins was established, we must always remember that there is a hereditary unit, which is a gene, which is actually a DNA portion. And that hereditary unit is responsible for encoding an intermediate molecule, which is called as messenger RNA. And that messenger RNA message is actually translated into protein in the next step. This process of decoding the genetic information, which is there in the blueprint, into protein, this entire process is referred to as gene expression. Is this fine? Should we go ahead from this point? Are you having any confusion or any doubts right at the moment? Or is it okay to move ahead from this point? Yes? Any doubts or problems that you are having? Okay, I got some responses. Okay, so should we go ahead from this point? Fine. So now there are a few fundamental questions that I'm going to put up in front of you, because we already know that this is how the scheme goes like. We already know everything about it. The relationship between genes and proteins, how it was established. That was done by the study that was conducted by Beadle and Tatum, who were the first one to give this association of genes with proteins, because proteins, we know now, are the executioners of the genetic information. How that information is actually relayed from the DNA to the protein, to establish this, you need to have a previous foundation which talks about that there is some relation between genes and proteins, and you need to prove that, right? That was done by Beadle and Tatum in their studies with orange bread mold, that is popularly referred to as Neurospora. So the study was done on Neurospora crassa. But there are certain things that we need to focus on uh, before we move ahead from this point. Okay, one important thing that one needs to understand is that it is not always like this. That means this central dogma of molecular biology is not always like this. Now we know that there is a process which is called as reverse transcription also possible, where the RNA is the information bearer and it is responsible for converting itself into DNA. This happens in retroviruses and like HIV. The process is called as reverse transcription. Also, we know that proteins are responsible for synthesis of DNA as well as RNA, because proteins play an important role in catalyzing this mechanism. So it's not always like this. So there are certain exceptions here, but as we say, exceptions prove the rule. So we'll consider this at this moment, as we have established the points that okay, that DNA is the genetic material. Now the information in the DNA has some relation with the protein that we need to establish, and that was done by the studies conducted by, they conducted a very well planned and a very broad scale studies that were done by Beadle and Tatum, and that was done in 1940s. You can imagine how tedious that job must be. Now, to understand their studies, that you need to understand a bit about how the fungal life cycle is. In case of Neurospora crassa, there it can go by both ways. That means it can divide vegetatively as well as, like for example, it is given over here. It can vegetatively divide, or it can undergo asexual reproduction, or it can also undergo sexual reproduction, wherein there is formation of two gametes which are named as A mating type and a small A mating type. Okay. Now, whenever these two gametes meet, then either of the two gametes can again lead to the formation of an organism as a whole, as a haploid organism, or the organisms might form, they might undergo a sexual reproduction in order to form gametes again. And this is the process. Okay, I'm not going to any details of it, but ultimately you get a pod like structure which is here referred to as ascus. Okay, which harbors the what you can say, the spores, which are referred to as ascospores. And each spore can be, that means 50% of those spores are of A type and 50% are of small a type. And individually they can germinate and give rise to the entire organism, which can further lead to formation of cells that fuse together and get to the formation of ascus again. So that's the story about Neurospora crassa life cycle. What they did was quite interesting. They use this ascus, wherein you have 50% of A type, capital A type, and 50% small a type, and they irradiated this ascus which with ultraviolet radiations or X-rays. Okay, now what you can say character of X-rays or ultraviolet radiations? Yes, any idea about this? Yes, any idea? What is the nature of these X-rays or ultraviolet radiations? What they must be doing? They are mutagenic, right? They can bring about mutations. So the concept is like when you irradiate the ascus with ultraviolet rays or X-rays, then there is a chance that these ascospores might undergo mutations. And then what they did was that they separated each of the ascospore from the ascus, and then they plated it onto a fungal growth medium, which is basically PDA, that is potato dextrose agar. Now, when you are putting up, say, spores here, then there was an interesting stuff they did. They used this media devoid of one amino acid, say histidine. Okay. Now, if the fungus is good enough to survive without histidine, then it can be considered as wild type. However, if it requires histidine in the medium for its growth, then it will not grow. That means the spore will remain as a spore, it will never germinate. Okay, so say suppose this is the spore which cannot germinate, then how do you do it? You go for another plate with PDA and you transfer this spore from this plate to this plate, which is with histidine. Okay, now if this grows, then you know that okay, this is the spore which was lacking the potential to grow if there is no histidine in the medium. So it is having a problem of production of histidine all by itself. Does that make sense? Yes.
Yes, sir.
Okay. Now what is understandable here is that you have converted, what you have done, you have converted the wild type into a mutant, right? By using mutagenic agents which are capable of targeting what? They are capable of targeting DNA. But that DNA, once targeted by the mutagenic agent, is responsible for somehow relaying that mutation into protein. And then because there is a mutation in the DNA, there is a defective protein that you are getting, which previously was able to, in the wild type, it was previously able to produce histidine all by itself and hence didn't require histidine in the medium, histidine supplementation. But now you have created a mutant which has a defect in the DNA and hence cannot produce a properly functional protein. So the organism cannot germinate on histidine devoid medium, but it will require histidine supplementation in the medium to germinate because it is not able to synthesize the protein which is responsible for histidine production. Does that make sense? I have given you just one example. They did it with almost every amino acid just to find out that, okay, in the minimal medium, this is called as a minimal medium in which you don't supplement most of the nutrients, okay, and in which you actually carry out germination, it will be referred to as a minimal medium. A minimal medium is something that will not have essential precursors. It will have only organic composition. So it is given over here. You have organic composition. Okay. So while doing this, what they are doing is that they are trying to pinpoint and find out that whether DNA mutation is related somehow with the organism's ability to synthesize certain components. Consider histidine here. Is it okay? Does that make sense to everyone? Yes or no? On this basis of observation, they put up a hypothesis, which was called as one gene, one enzyme hypothesis. Later on, however, it was disapproved because we will talk about this in greater detail. It's not a correct one, but to some extent, they established this relationship that the gene is responsible for synthesis of an enzyme, and the enzymes are the ones which are going to be responsible for making the organism self-sufficient to survive in a minimal media. Does that make sense? Okay, is it okay, or you are having any doubts here? If you are having any doubts, you can ask me, because you are nearing to the end of the class. So in case you are having any doubts, okay, what I will do is that I will stop here and let you people ask questions regarding this experimental setup if you are having any doubts here. But as of now, is it okay if I say that after the what you can say, after the hypothesis that was formulated by Archibald Garrett, the experimental evidence that was given came up much later, almost 40 years after Archibald Garrett, and the experimental evidence that established a relation between DNA and protein was coming from the experiments that were conducted on Neurospora crassa by Beadle and Tatum. So Beadle and Tatum are given all the credits of establishing a relationship between genes and proteins. So if you are having any doubts, you can ask me.
Sir, why spores were used and not
The entire organism, because see, spores are the ones which are going to get germinated into an entire organism. So if there is a mutation in the spore, which is mostly a haploid spore, if there is a mutation there, the mutation will be carried in entire organism, right? However, if you irradiate the organism, then it could be a somatic kind of a mutation. That means because it's a multicellular organism, it can be a mutation only in one of the two cells, and not the entire organism. So when it germinates, or when it actually rises rise to the cells, which the mating types, which are going to meet together in order to form a diploid, what you can say, um, uh, diploid organism, which is further going to give rise to haploid spores. That will not happen. Does that make sense?
Yes, yes, sir. But um,
Yes. So the problem was like why he is irradiating the spores, right? The spores are going to germinate into a haploid organism. You can see that there is N written over here. It's a haploid organism.
yes,
Haploid
sir.
organism gives rise to haploid cells which fuse together, which are referred to as mating types, which fuse together in order to give rise to a diploid system, which further undergoes meiosis and gives rise to 8 spore condition. Each of them are haploid, out of which 50% are of A type and 50% are of small a type.
Yes, sir. My problem is not, I mean, irradiating the spores, but
Right.
uh, it is that why are we checking in spores for the production of
No, it is going to
amino
germinate
acids? Why
into,
not in this?
right. It is going to germinate into the entire organism, right? It is haploid organism. So if it germinates, see, you are in 1940s, right? So you will have to have some mechanism by the way of which you can track whether the spores are capable of germinating or not, right? So if it can germinate in and form the entire organism, even on the minimal media, then it is capable, it is like wild type. It is capable of synthesizing all necessary components which are responsible for germination, and it is capable of dividing, right? But if it
Yes,
cannot germinate, then I can very easily pick up that spore and transfer it onto another medium and find out that is it. Now just imagine this. You have minimal medium with you. You are beginning with minimal media. There is nothing there in the medium. It has only inorganic nitrogen, some salts, sugar as the carbon source, and biotin as one of the vitamins, meaning that's one just addition. Now, in case you are studying, say, vitamin deficiency, then that can also be studied over here. I'm giving another example, however. Okay, so just imagine that you have a minimal medium which consists of only sugar, salts, and inorganic nitrogen. Using these very simple building blocks, can you synthesize and germinate into the complete organism on this minimal media? If you can, you are a wild type. If you cannot, then I can very easily pick up a spore and transfer it into another plate which contains minimal media that is supplemented with some amino acids, say histidine. If it can germinate here, I know that here it was facing a problem because of no histidine, but here it is not having a problem because I have supplemented it with histidine. So I know that there is problem in histidine synthesis pathway. Is this fine?
yes, sir.
Right? This is a very difficult thing to do, however. Just imagine that you have a spore that is not germinating. You have 20 different plates with you, each supplemented with only one amino acid. You put it onto histidine plate, it doesn't grow. You put it onto phenylalanine plate, it doesn't grow. You put it on tyrosine plate, it grows. What it means is that it is having a defect in tyrosine synthesis path. Am I making sense?
Yes, sir.
So that is how they figured out that, okay, there is a problem here in this mechanism, in this pathway. I hope that the point is clear.
Yes, sir. But again, one doubt comes in my mind is that when these uh, capital A nucleus and small a nucleus
Yeah,
they fuse, they
right.
might show some different characteristic.
Right, obviously. See, but once you are having, say, a histidine mutant here, imagine this is a histidine mutant. You are not using a mutation to germinate, right? You are using wild types only. You are using wild types to synthesize the spores, which are wild type spores, according to you. and now you are irradiating it with ultraviolet radiations. You are not allowing a mutant to meet a mutant. Is this making sense? Because that will create problems for you later on. You 100% know, okay, those spores which germinate on the minimal media, do you agree with me that they are wild type?
Yes, sir.
Can I not use the cells from these wild types to meet together?
Yes, sir.
So they will be all self-sufficient to give rise to spores which are self-sufficient,
Yeah, right.
right? Now I'm irradiating them. So what's the problem?
Okay.
I hope that the point is clear now.
Yes, sir. Yes, sir.